然后的话呢，非常欢迎大家来到我们今天这样的一个AHS 的这样的一个论坛然后的话呢，下面的部分由于我们是要有不同的国际的听众下面的部分我们会以这个英文来进行这个主持，包括后面田教授的这样的一个报告。Okay, so good morning, everyone. So from the online platforms, and we are delighted to have you on this AHS forum for this session. And today we are delighted to invite uh, Professor Tian Xiaoyu from the Chinese University of Hong Kong. So please let me first give a brief introduction about Professor Tian.
Okay, I have one.
Okay. So, Mr. Tian is now an associate professor of the School of Biomedical Science at the Chinese University of Hong Kong. And she graduated from the Chinese University of Hong Kong and then got pursued the postdoc studies in America and then now come back to Hong Kong for this continued research. And she is now focusing on the research of vascular homeostasis, inflammation, atherosclerosis, and endothelial cell function. And she has published a lot of papers with a very high H index. And today, she will give us a talk about the role of mitochondrial metabolism in endothelial cell dysfunction and atherosclerosis. So now I will give to Professor Tian and to her for the next session. So, Professor Tian, please.
So, thank you, Professor Zhang. So, hello everyone. So, I hope there's no problem to see my screen, right? And hear me on the Zoom. So,
Yeah.
because we probably have some international participants, so the talk we will be delivered in English. So, but if you have any questions after the talk, you feel free to ask in any kind of language. Today, we will be talking about mainly the vascular biology and atherosclerosis, and with a little bit of background of our previous research. So I work in the Chinese University of Hong Kong. So first of all, I want to give you a little bit of introduction on what is atherosclerosis and why we think the endothelial cell function is important, and why the dysfunction of the endothelial cells could lead to this very important disease. Because if there's a problem with your blood vessels in the heart, in the brain, or any kind of organs, then it will cause ischemia and organ damage. So why we think endothelial cell is important is that if we look at this, let me use the laser pointer. So if we look at this vessel structure, so this is one of the thoracic aorta, and when it branches into probably the aortic arch, we can see that the blood flow will have some disturbance, right? Some we call the oscillatory blood flow or disturbed blood flow. And this kind of change of the flow patterns can induce a series of change in the endothelial cells, because endothelial cells is a cell that is directly exposed to this kind of stress. And we can see that when in the straight part of the vessel, the endothelial cells aligns very well. And also the structure is very nice, and there's not much of inflammation as indicated by one of the markers in green. This is one of the adhesion molecules. And if we look at those areas that is exposed to the disturbed flow, and we can see that the cells doesn't really align well. So the red color aligns the indicate the membrane of the endothelial cells. And also we can see a lot of increase of the inflammatory markers, these adhesion molecules on the surface of the cells. And one important thing is that if you do exercise, you can actually increase the shear stress, the laminar shear stress in these vessels, and then change the flow pattern. So that is why doing exercise is really important to maintain the vascular health. So since a long time ago, since my PhD time until now, so we have been working a lot on measuring the endothelial cell function. And one of the experiments that we commonly do is to measure the tension in the isolated blood vessels using these two types of tension measurement. So for example, if we isolate one of the small vessels from the vasculature, and we can put it under a pressurized condition, And then if we infuse in the buffer, and then we can measure the diameter of this vessel, so using a microscope. And then we can test, for example, the response of this vessel to different pressure or to vasoconstrictors and vasodilators. So, for example, taking this mesenteric arteries from here. And we can also measure the isometric tension by just putting this vessel segment between the wires, and then to stretch it, and then we have tension. And if you add a vasoactive substance, then you can measure the tension change. So we use mainly these two methods to measure the vascular tension. And in addition to that, we can also use confocal microscopy to measure the calcium. So using a time-lapse method to measure the calcium movement, for example, in this cerebral vessels in the isolated from the brain of the rat. So Professor Zhao, so sometimes I have probably have a bad connection. So if that happens, then I maybe like pop out for a few seconds. So just bear with me if that happens.
Okay.
Okay. So that is the vascular biology that we are interested, and also to do research, we also have need to look for some signaling or pathway or some molecules, right? So that's what everybody does, and we are particularly interested in the communication between the nucleus and the mitochondria, and we also call it like mitonuclear signaling, and it includes various of response. So for example, the nucleus, when it senses a cellular stress, it can tell the nucleus to start a transcription or start to have some epigenetic change, so that the mitochondrial proteins will be adapted to this kind of stress. So using this kind of anterograde signaling pathways. So within these pathways, we have actually worked on a bit of this and that. So for example, we measured the mitochondrial antioxidant response. So by looking at the mitochondrial ROS production, the reactive oxygen species production. So in the endothelial cells, we can either measure it in the isolated cells in a time-lapse imaging method, or we can also take out the blood vessel and then directly measure the mitochondrial ROS production from the endothelium by just imaging the endothelial layer from these isolated vessels. And here we show that, so if we give the mice a high-fat diet, so it becomes fat, and we can see a lot of mitochondrial ROS production, so compared to the lean mice. And if we knock out one of the key antioxidant mitochondrial enzymes in these mice, we can see the mitochondrial ROS production actually increases at the basal level, and also it further increases in the mice from which was fed on the high-fat diet. So this was the method that we helped to develop, and then a lot of labs are also using this kind of method now. So within this anterograde signaling of the mitochondria to nucleus communication, so we are also we also did a little bit on one of the key transcription factor, which is PPARD, because it mediates a lot of the gene transcriptions that are involved in the red dot response in the vascular cells. So this is one nuclear receptor that is ubiquitously expressed in all the cells, and in particular, the vasculature highly express this particular subtype of the nuclear receptor. And from long time ago, there are papers that show the physical exercise actually the effect of physical exercise actually is mediated largely through this nuclear receptor. And if you give a agonist to activate or a ligand to activate this PPL receptor, then it can actually mimic the beneficial effect of exercise. And that work was done a long time ago, mainly in the muscles and in other metabolic organs. So for on our side, so we mainly focused on the effect of this transcription factor in the vasculature, and what we initially showed is that, for example, if we gave the fat mice, the diabetic fat mice, so this one is, we call it DBDB here, it's the leptin receptor knockout mice, and we can see that so we can see that, we gave these mice exercise, so we put them on the treadmill, and you can see these little fat guys run, and they will do this exercise 5 days a week. So that's because our graduate student also need to rest on Saturdays and Sundays, so the mice also do not run on the weekends. And after, like, let's say 4 weeks or 8 weeks of this kind of physical exercise, then we can see that if we measure the flow-mediated vasodilation in the mesenteric arteries from these fat mice, we can see that the flow-dependent vasodilation actually improve a lot. So after they perform this exercise for a few weeks. And very importantly, we also find that this kind of improvement is largely mediated by the PPL delta nuclear receptor. And this showed here is a endothelium-dependent vasodilation, and this is one of the direct measurement of the endothelial function. So if the tension reduces a lot, then it means that the vasodilation is being improved. So what we can see here is that if the mice is given high-fat diet, then the vasodilation is reduced, and if the mice receive the exercise training, the EX group here, then the vasodilation improve a lot. And this effect is actually gone if the mice doesn't have the PPL delta. So this also demonstrates that the effect of physical exercise on the blood vessels is also largely mediated, by the nuclear receptor PPL delta. So I didn't show the details here, but what we did is that to show the PPL delta, if it is activated, then it reduces various kind of stress signals, including the ROS and also the unfolded protein response. And the outcome is that you have more nitric oxide production from the blood vessels, so the endothelial cell function kind of improved. So that is our previous work mainly on the anterograde signaling as part of this nuclear to mitochondrial communication process. And in addition, you can see that this kind of communication also include various other responses. And also, we also want to introduce to you a little bit of our work on the mitonuclear feedback response. So this means that if there is any stress response, mainly by the unfolded protein response and other proteolytic stress. So this kind of stress can be sensed by the mitochondrial enzymes. And after that, then the mitochondrial enzymes may be able to transmit the signal through the nuclear transcription, and then to modulate and make the mitochondria adapt better to the stress response. So this is what we call the mitonuclear feedback response. And among them, we are particularly interested in this enzyme. So because this enzyme is a deacetylase, and as we know that there are a bunch of different citrulline, right? And then some of them are located in the nucleus, and what they do is to mainly do the histone deacetylation and to modulate the epigenetic response. So in addition to that response, mainly mediated by SIRT1, you also have sirtuin 6 and 4 and 5. So they do a little bit of different epigenetic modulation in addition to the deacetylation. And in particular, this sirtuin 3 is actually not located in the nucleus, but it is mainly localized in the mitochondria. And what it does is to deacetylate the mainly the mitochondrial enzymes, and a lot of these enzymes are actually involved in the mitochondrial metabolism. And in the past, there have been some work which showed that in the cardiovascular disease, a lot of these risk factors, so including the inflammatory signals and the high lipid, high glucose, and angiotensin too. So these kind of risk factors can actually impair or reduce the sirtuin 3 expression. And then you have an increased mitochondrial oxidative stress, and that can lead to a various of cellular responses and then lead to a vascular dysfunction and has a really important role in mediating hypertension in the mice. And also, as the sirtuin 1 and 3, this kind of enzyme, they will need a substrate to work, right? So you need to give them a substrate, otherwise this enzymes doesn't work. And the substrate that is important for maintaining its activity is the NAD, the nicotinamide adenine dinucleotide NAD. And the NAD would actually serve as the substrate so that it will help to maintain the acetylation mediated by sirtuin 1 and sirtuin 3. So let's say if you give the cell more NAD, it could probably enhance the activity of these sirtuins and then maybe have a beneficial effect. And this is what we did with the NAD and one of its precursors. So the precursor is called nicotinamide riboside. And if you give the cells more of this NR, it would also increase the biosynthesis of NAD, and which will serve as a substrate. So if we give the cells or the mice this NR, and you can actually get this from the local pharmacy because they sell it as a nutritional supplement now, and there seems to be a very big market on selling this kind of NAD-related supplement, right? So for this particular experiment, we gave the mice a treatment of the nicotinamide riboside every day. And then after 2 weeks, we measure the atherosclerosis. So in these ApoE knockout mice. And these mice, they would develop the atherosclerotic plaque, as we can see by the white color here. So white color indicate the atherosclerotic plaque formation and upon the feeding of the high cholesterol diet. And we can see that after a very short treatment, like for 2 weeks, then we can see actually the plaque formation reduced a lot in this aortic arch and also the branches of the aortic arch in the ApoE knockout mice. And this can also be seen in the aortic root section. So this is the aortic valve. We can see 3 valves here. And this is one area that we use for this kind of measurement to show the effect of any drugs or gene effect on atherosclerosis. And we can see the red color indicate the plaque formation, and it really reduces a lot after the mice receive this nicotinamide riboside treatment. And in addition to that, we can also see that the endothelial cell function indicated by the vasodilation response also improve a lot in the mice that receive the treatment. So in fact, so if we look at the NAD's effect in the cells, in the vascular endothelial cells, we can see that if we give the cells an inflammatory stimuli, it would impair the mitochondrial metabolism as measured by the seahorse assay, right? So if you use this seahorse assay to measure the OCAR, you will see that the cytokines actually impaired the mitochondrial metabolism. And if we give it a supplement of NAD, it would rescue the mitochondrial dysfunction. And in the cells that without the sirtuin 3, we can see that the effect of the cytokines is still there. And in fact, so the NAD can improve the mitochondrial function whether the cells have sirtuin 3 or doesn't have sirtuin 3. So it has effect independent of sirtuin 3 as well. And in the blood vessels, we found that, so although in the previous cell experiment, we found that the effect of NAD is not only dependent on sirtuin 3, But if we look at the endothelial function, so mainly mediated by a nitric oxide induced vasodilation in the blood vessels, we can see that it is again reduced by the cytokine IL-1 beta. And if we use NAD, it improved a lot and rescued the vasodilation. But if the mice, they don't have sirtuin 3, we can see that the cytokine still works, so impaired the vasodilation a lot, but the effect of NAD is not there. So this gives us some hint of a role of sirtuin 3 may be important for making nitric oxide. And it has probably nothing to do with the effect of NAD on the mitochondrial function, but probably have something to do with making nitric oxide. And then this inspires us to further study how it could be involved in making nitric oxide. And because the nitric oxide and endothelial function, as I mentioned earlier, are really important for reducing the vascular inflammation and atherosclerosis formation. So what we did is that we used this sirtuin 3 endothelial selective knockout mice. So if we see the ECKO means that these mice, they don't have sirtuin 3 in the endothelial cells. And we can see that if we use a method to induce hyperlipidemia by overexpress a gene. This is an enzyme to degrade the LDL receptor in the liver. And then if we feed the mice with high cholesterol diet, then we would have very high hyper or cholesterol millibare in the blood. And that can induce the atherosclerosis formation in the mice. And if we use this, then we can see that in the mice without sirtuin 3, you can see a lot more atherosclerotic plaque formation. So as indicated by this red color here in the blood vessel. So this is the entire blood vessel, aortic arches on the top, and then you have a thoracic aorta and then some branches. And we also see that this kind of plaque mostly form in the branching areas and also in the curvatures. And then, in fact, if we detect the plaque formation in the aortic root, we can see a lot more. And also, the collagen is less in these plaque areas, which is an indication of a phenotype of we call unstable plaque. So this means that these plaques are more dangerous and they are more prone to a rupture, and then a rupture would be very dangerous because it can cause thrombosis and embolism, and then can block the blood vessels. So if we look at these plaque areas in a little more details, we can also see that in the mice without sirtuin 3, we can detect a lot more vascular inflammation as showed by these adhesion molecules, and also more infiltration of the vascular macrophages as indicated by the CD68 here. So this is an indication of the phenotype of increased atherosclerosis may be also associated with more vascular inflammation. And in fact, so we want to understand why this sirtuin 3 is important for endothelial function, right? So as we mentioned earlier, that this one is an enzyme and is a protein deacetylase. So what it does is to deacetylate the protein so that you can detect by the acetylated lysine to see whether these proteins are deacetylated. And also it is located in the mitochondria. So if we do a localization, we can see that, in fact, we can see a lot of the sirtuin 3 expression in the mitochondrial area, but you can also see a little bit of sirtuin 3 expression in the nucleus and in the cytosol. This is because they are actually a full-length and a cleaved active form of sirtuin 3, and that's why you can also detect them in other cellular compartments as well. But mostly it is enriched in the mitochondria. And because it mediates the deacetylation, so if we detect the acetylated lysine, so which is indicated by this ACK here, so we can see that in the endothelial cells taken from these mice, we can see more acetylation as showed by this ACK signal here. So these mice, they don't have Saturn3 in the endothelial cells, and they have a hyperacetylation of a bunch of different proteins. And if we detect the acetylation in the mitochondrial fraction from a cell line, so this is a mouse endothelial cell line, we can also see that the cells with a knockdown of Saturn3, they have hyperacetylation of the mitochondrial protein. So this validated that, so this Saturn3 is actually important for maintaining the deacetylation and activity of the proteins. And without it, then you have a hyperacetylation of many different proteins. So from this experiment, so naturally we would think that the next is to detect, so what are the identity of these hyperacetylated proteins, right? So this is what we normally do. So after this, then you run a gel, and then you may cut the gel and then to detect the proteins by MS, or you can run a proteomics to detect different kind of proteins. And this is actually what we did, but so I will go through this one by one. So actually we did this, but then we found that if we purified the mitochondrial fraction and run the proteomics to detect the acetylated proteins, so we do have some target. So we have some of the proteins, or if we detect the acetylated sites, so we also have a bunch of different candidates. But it turns out that if we look at these differentially expressed acetylated proteins, they are not actually involved in the mitochondrial function. So we can see that we have a lot of like non-specific signals from other cellular response, so that are not related to the mitochondrial function. So after this experiment, we wondered why. So one possibility is that the protein fraction wasn't really very purified to contain most of the mitochondrial fraction. It probably contained a little bit of cytosolic proteins as well. And another guess is that just the difference between the wild type cells and the knockout cells are not really that big. So that's why we did not detect any meaningful differentially expressed protein. So after running this omics. So after this, so my student and I were like sitting on this data for a really long time. And then I kind of like give my student a very subtle signal, try to ask her to do this essay again. And it looks like she doesn't want to do it. So we took a long time to try to figure out like what is wrong, and then we were like running out of budget to run this again. So we have been sitting on this data for a long time, and then from time to time I dig this out and then try to look at it, and then still find that the data doesn't make any sense. And then after that, so we still want my student need to finish her PhD, and then we also need the project to be finished. So we need to figure out a way, right? So to really find out a solution to this problem. So what we did is that we actually went back to the data set that was performed by other researchers on the liver cells. So this is actually not the endothelial cells. And this is from a few papers, which actually what they detect is the hepatocyte from the wild type and the knockout mice. And then from these two papers, so our reason is that if the Saturn3 is important for mitochondrial protein acetylation, it would be universal for not only endothelial cells, but other cells as well, right? So one of the cells that is heavily regulated by Saturn3 will be the liver. So this is what people have done in the liver. And out of this data, we found that there are actually some of the enzymes that are involved in endothelial function. And one of the enzymes that we found from this data set is called the argininosuccinate synthase 1, so ASS1 here. So this is detected in the liver. And why this enzyme is important is because is because of is because this is one enzyme that would actually recycle the citrulline back to arginine. And why arginine is important is because arginine is the substrate for the nitric oxide synthase to make nitric oxide. So if you don't have enough L-arginine, it is possible that the nitric oxide production will be reduced. So um, this was actually inspired by an early work which I contributed and was done in Baylor by Professor Brandon Lee. So at that time, what they did is that they found an arginino succinate lyase, so ASL, and ASL is actually not directly related to the endothelial function. So why this gene is important is because the ASL, it is heavily expressed in the kidney and involved in the urea cycle because urea cycle would be on the other side of this pathway. And accidentally, what Professor Lee's group found is that the patient without ASL, they also have hypertension, and then they want to understand why this genetic disease is associated with hypertension. And what they did is that they did a knockout of ASL in the endothelial cells, and then found these mice, they would have reduced nitric oxide production and reduced endothelium-dependent nitric oxide-mediated vasodilation, and also these mice would have high blood pressure as well. So this is an indication of actually some enzymes that are involved in making L-arginine may be important for making nitric oxide. And without these enzymes, then you may not have enough L-arginine to make nitric oxide. So in fact, in the proteomic data, we try to see whether ASL and ASS1 are both modulated by sirtuin 3. And in fact, we didn't see any change with ASL. So that is why we only find a significant change with ASS1. And so we identified this target from others that done it in the liver cells. So we confirmed that this enzyme, this ASS1, is actually hyperacetylated in the endothelial cells without sirtuin 3. So we can see that if we run a immunoprecipitation, we can see that the hyperacetylated proteins contain a lot of ASS1. And then if we run a immunoprecipitation on the other way, so whether either we pull down ASS1 or if we pull down the acetylated lysine, so it gives you a same result to show a hyperacetylation of ASS1, so in the cells without sirtuin 3. And in fact, this ASS1 is also closely associated with the mitochondria, so it is not exactly within the mitochondria, but it is enriched in the mitochondria, and some of them are actually detected outside, but in close proximity. And after this, so we confirm that sirtuin 3 can actually deacetylize SS1. And then we also want to know a little more on which is the interaction site on the ASS1 that is mediated by sirtuin 3. So we did this assay to mutate two of the sites that are important for maintaining the activity of maintaining the activity of SS1, and also they can be acetylated. So if we mutate the lysine to another amino acid, then it will not be able to get deacetylated by sirtuin 3. So if we see here, then we can see that with sirtuin 3 expression, then the acetylation of the ASOS1 is reduced. And with the mutation, then we cannot see any effect of this sirtuin 3 expression. So indicate that these two sites may be the major site that is deacetylated by SS1 on the that is deacetylated, sorry, by sirtuin 3 on the SS1, and may be important for maintaining the enzymatic activity of SS1, which would make more L-arginine. And the next question we want to really answer is that whether SS1 would mediate L-arginine synthesis, right? So using the same wild-type cell and the mutant cell, we can see that in the wild-type cell, so if you give it more sirtuin-3, we can see that if the ASS1 is deacetylated, then the cells start to produce more L-arginine. And with the mutation, then you don't see much production of this L-arginine, so indicate that this site is important for the enzymatic activity. And very importantly is that in the cells, we can see that such ASS1 silenced, then we have less L-arginine production. And if we silence the sirtuin-3, then we also get less L-arginine production from the cells. And in the conditioned medium, then we detect a very similar reduction in the L-arginine production from the cells. And in the mice, so what we can see here is that, so if we compare the amino acid profile in the plasma from the wild-type mice and the knockout mice, we can see that all the other amino acid doesn't really change much. The only amino acid that change a lot is the L-arginine, so in the bottom. And we measure this by metabolomics, and we also measure it by enzymatic assays. So this is the metabolomics result interpretation, which is the same as the graph. And if we use other like ELISA method to measure L-arginine, we can also see a reduction of L-arginine concentration in the blood plasma. So this is a actually to us, this is kind of like an important finding, is because in the past we know that L-arginine is actually not an essential amino acid, and in fact, the endothelial cells does make a lot of L-arginine. So if the endothelial cells doesn't make arginine, you actually have less L-arginine in the bloodstream. So that makes a lot of difference. And then we validated this effect of L-arginine on the blood vessels. So what we see is that in the mice, in the isolated blood vessels from the mice, if we use the cytokine to impair its vasodilation, which is mediated by nitric oxide. And if we give it back the L-arginine, it can rescue the endothelial function. And this L-arginine also works in the knockout mice because the knockout mice doesn't have that much of L-arginine. And not only the nitric oxide production, but also if we look at the mitochondrial ROS production, this L-arginine supplement can also reduce a lot of the ROS production in the cells, either with the knockdown of sirtuin-3 or with the knockdown of SS1 as well. So if we look at the mitochondrial metabolism, we can also see that if we give the cells back the L-arginine, then we can see a lot of improvement of the mitochondrial function, so indicate by the seahorse assay. So in both the sirtuin-3 and AS1 deletion mice, then in the knockdown cells, then we have a impaired mitochondrial function, which is rescued by the L-arginine supplementation. So the next question that we want to ask is that if L-arginine is really important for making nitric oxide to improve mitochondrial function, so does it really work like in the mice, right, to reduce the atherosclerosis formation? So if we give the mice an L-arginine supplement in the everyday, so for a few weeks, so what we see is that without the L-arginine, you have a lot more plaque formation. So especially in the knockout mice, and with the supplement of L-arginine, you do see a lot of improvement. So and what we also see here is that the concentration of L-arginine in the blood reduce a lot in the knockout mice, and then if you give it supplement, then it comes back, and the lesion measurement is showed the same as the images. And if we look at the plaque phenotype, we can also see that not only the plaque size reduces, but also the plaque become more stable. So you have more fibrotic marker, the alpha smooth muscle acting on the surface of the plaque, so indicating that this is a more stable plaque, so instead of a unstable plaque that is prone to rupture. And in fact, if we give the mice L-citrulline, the precursor of ASS1, it doesn't really work. So we also have that data, but there's not much time for me to show here. So this is what we finding is these mice is that in fact the citrulline 3 is really important for maintaining the deacetylation of this very critical enzyme in making L-arginine. And in the body, the L-arginine, a lot of them are actually made by the endothelial cell. So although it is a semi-essential amino acid, and without L-arginine, so you don't have enough nitric oxide production. And this nitric oxide production is not only important for vasodilation, but also very important for maintaining mitochondrial function and to reduce vascular inflammation as well. So if you give back L-arginine, then it can improve all the phenotypes that we observe in the knockout mice. So this is our little bit of work like here and there related to the anterograde signaling from the nucleus to the mitochondria and a little bit of mitonuclear feedback. So mainly focus on the citrulline 3, this mitochondrial deacetylase. And we also have some ongoing work that is related to the retrograde signaling of this pathway, but maybe I can talk to you more about that next time. So when we are more ready. So in the end, I would like to thank all my previous supervisors and collaborators from both from the CUHK and also from other institutions. So including my PhD supervisor, Professor Huan Yu, and our collaborator in the sports science department, Professor Stephen Wang, and also biomedical engineering colleagues, Jonathan and Kathy, the vascular biologist, and Dr. Kong from endocrine department, and also Professor Johan Owerth, who kindly provided us with the citrulline flux mice, and Dr. Anna Jan, who is now in Macau, and she contributed a lot to the PPLD story at the beginning, and also our collaborator at UCSD for giving us some precious reagents as well. And also very importantly, the PhD student and the research staff who contributed to this work. So many the PhD student who did the citrulline 3 story during her PhD, and other people in the lab, and some of the undergraduate students as well. And also I want to mention that we have PhD quota to admit student in the next cycle, so who will be admitted in 2024 August, and also we have quota for admitting student in 2025 as well. So if any student, you are welcome to contact me for more discussion on if you want to do a PhD in CHK. And our funding support mainly from Hong Kong Research Grant Council, and also a little bit from NSFC, and also from the Department of Health, the Health and Medical Research Funding as well. So I would like to also thank the audience on the Zoom and also on different platforms as well. So questions are welcome.
Okay, so thanks, Professor Tian, for this excellent and inspiring work. Now we welcome our questions from all the platforms and all the audience in this Zoom meeting. 好，非常感谢田教授刚才精彩的报告啊。后面的话，我们的这个问题的话呢，就是可以英文也可以中文的形式来进行。OK。Okay, maybe start from my question. It's about a more general question. So, I don't know if you have ever checked out what happened to the brain vessels, the cerebrovascular functions. I think it's something different between the heart and the brain vessels, because for the brain vessels, it's not only for this endothelial cells, but also it calls for the brain blood barrier and also other glial cells in this. So can you give any like comments or suggestions on this? Because now we are also trying to work on some projects on this endothelial cells in the cerebrovascular functions. Thank you.
Thank you. Thank you, Professor Zhang. So, that's a really meaningful question. So, I don't think I have a good answer for you. So, based on what I know is that the brain vessels. So, first of all, the microvasculature is very different. So, you have a blood-brain barrier. So, you have a lot of those junctional proteins, and another thing is for the cerebral blood vessel. So, this is the major site if you have ischemia injury, right? And in fact, so in the I think from a while ago, we tried to study the brain vessels and try to use the injury models. And in fact, we don't have any manpower to do it. And also we really don't have any student to look at the brain vessels. So I believe this enzyme, so other people may have done it in stroke models or other things as well. So, but from our side, we haven't really looked for anything related to that yet. Yeah, we do have other models to study the microvascular, so especially the capillary endothelial cells. So in other organs. So at the moment, we don't really have the capacity to study the brain vessels. Yeah. Yeah, it's probably involved in a way, and because the brain vessels is very different. So yeah,
Okay.
that's a meaningful question. So I hope that in the future we have the capacity to expand a little bit to do the cerebral vasculature as well.
Yeah. I think because cerebral.
Do you still remember Owen Gao Hao? So he was with Professor Yong for a while, right? So at that time we talked about doing something related to brain endothelial cells and parasite, but then we don't really have the capacity to do everything.
Yeah, right.
Okay. Thanks, Professor Tian, and gave the wonderful talk. So I have 2 or 3 questions. The first one is you mentioned about the 33 and have the like the impact the EC function, and do you think whether have the effect to the lip drop lysis or not? How to correlation with the lip drop lysis?
Lipolysis. The lipid, right?
Yeah.
So as Professor Chen, that's a very important question, because I asked my student to do something related to the lipid and cholesterol within the endothelial cells, and then we do see that with the expression of sirtuin 3, with or without it, there's some difference. Yeah. So our idea is that if you have more free cholesterol in the cytosol, in the cytosol of the endothelial cells, then it can cause some toxic response and, for example, inflammatory response. So, but we are really very preliminary for that part of the influence on the lipid metabolism.
And also the second question, and you also mentioned about the carcin concentrate, the carcin. Did you make some research about the carcin dependent protein and how to regulate? Like later you also talk about the ACS1 and whether have this relationship.
Which concentration?
The carcin dependent protein.
哪一个？
，Yeah，
哪一个？
carson，钙条蛋白
盖球carmodulin 那个。
，Yeah，Yeah，Yeah，Yeah，Yeah。
No, we didn't really take too 就是我们那个没有特别做多少，因为我们只是会用那个钙的信号来做一个就是它
Yes,
在
carson
内
imaging。
对它在内皮细胞和在平滑肌细胞都可以用calcium imaging 来做那个就是钙的信号来做一个。
But I think and currently so many carson dependent protein and can
对对对，就是
regulate。
但是我们没有去详细的做这些 calcineurin 和就是 calmodulin 这些的 signaling，就是这些的信号通路我们没有就是做的非常多。
Mm.
嗯
Another question, and like the because we do the exercise physical activity, so like the how to whether how to whether have the effect because mention about the blood flow, this is the physical function. So whether like the later you mention about the ACS1, this enzyme, like the physical activity or exercise, how to
嗯
effect it?
嗯嗯。Yes, so this is a really important question. So, in fact, so Professor Wang from the sports science department is actually trying to recruit some elderly to do an exercise project, and then maybe using those NAD related supplements. So, if let's say in the people, if you give them more NAD related substrate, or if you can uh, upregulate the sirtuin 3 or these or any molecules we involve in this pathway. So it maybe enhance the effect of exercise as well.
Okay,
这个
okay,
这个
thank you.
是就是对，就是这个问题非常好，就是因为我们暂时在老鼠上面我们就没有做这一部分，就是比如说它运动的效果是不是也受这个 protein 的介导？对，
Yeah, yeah,
对
yeah.
对对。但是我们有一些就是和别人合作的工作，他就想看一下，就是它下游也有一些和呃和那个线粒体的钙的信号有关的，它也是受这个可能是受这个 enzyme 的调控。但是
Okay,
就是运动的这一部分，我们就没有说再去详细的看它，比如说运动的 benefit 是不是受这个 enzyme 调控。但是我们
enzyme.
有一
Yeah.
些别的，就是我们有一些还没有做完的工作，就是它的运动的效率，还有它的运动的 endurance 是有一些别的，是在内皮细胞上面的作用。就可能下次如果有机会，做的比较完整的时候，可以再和大家 share 一下。
Okay, okay,
嗯，
thank you. I found the Professor Liu from the Wuhan University. 刘教授，刘教授在线吗？
可能走开了，嗯，有可能走开了。
Okay, so Professor Zhang, you
啊，
continue.
对，其实田教授可能请教一下，就是关于这个 acetylation，就是蛋白的 acetylation 的问题啊，因为我不是非常的明白这个，不是非常的懂这个领域。就是现在的话，acetylation 的话，它的这个acetylase 和 deacetylase 其实都还是比较清楚的，对吗？在这两 part 还是比较清楚的。然后的话呢，就是它这个东西，这个 acetylation 的话，它会不会它和我们身体的这个 metabolism、homeostasis 这个之间，它会有怎样的这样的一个关系呢？然后这个东西它跟我们做的这个 physical exercise 又会有怎样的一个联系呢？您可不可以简单的给我们简单的这个 briefing 一下这个东西？
这个就是因为我自己的了解是也非常的有限，就是我只能可能 comment on 我知道的东西，就是至少它这个 sirtuin 就是这几个 sirtuin，那么它 大部分都是这个乙酰化和去乙酰化，但是它有一些就是 Sirtuin 456，它是会有一些别的那个 protein 的 modification 的，就是像myelination，就是它会有别的一些那个我也不太记得它那个中文是什么，就是然后有很多的研究，他们是主要是集中在代谢上做，就是在肝脏脂肪和骨骼肌的这些，它因为它比较活跃，然后它如果有这些，就是它中间会有一些 metabolite 的 intermediate，它会直接去参与这些这些就是这些叫蛋白质的修饰，对吧？就是这些不同的修饰，对，它其实是有很多别的，就是主要做代谢的，他们就会做很多关于这些蛋白质修饰和代谢其中的某一些中间产物的关系。它其实有很多都是有联系的，但是就其实有非常多的这些代谢的中间产物都在参与这个过程。
嗯嗯嗯嗯，
嗯，
对，好，我们看一下，
好，
老师，包括我们这个几个直播平台上面，看一下有没有能够有没有反馈到
你
之前的留言。
直播平台的看，就是他们有没有那个，就是在留言框里面，就是看问一下他们那个主持直播平台的。
对，陈教授如果没有的话，那我们今天要不就先
嗯，好。
这个样子。
如果没有问题，我们今天就先到
好，
此吧。就是这个，也非常感谢，也非常感谢你们两个教授的辛苦的工作，一个田教授和
谢谢
张教授。
陈教授，
啊，
谢谢张教授。
谢谢。
再还有一个，你刚才谈到的那个 N2 treatment，那个
哎
N2是什么
哎，那就是 NMN，现在就
？AID 的那个吗？
现在很多地方都在
那个
卖的，对。
NID。
然后NAD、NAM，它就是有一些是前体
啊，
嘛，然后它好像不同的公司会做一些不同的，有的是 N 啊，有的是 NMN 这些，对，就现在
那你那个
现在很
2是代
多像
表什么呢？
我们这边就是
我刚才没有跟上。
是它就是一个其实我们可以去看一下刚才那个通路啊，我们还有时间，我们可以去看一下刚才那个通路。就是 R 是这个东西，就是 nicotinamide riboside，然后它这个 NR 和 NNM 都有很多公司做了一些 supplement，然后就
啊
是它是 nutritional supplement 嘛，因为它不是药，
啊
对，然
啊
后 NAD 就是它这个 immediate 的 substrate，呃，给这两个 enzyme 它是一个 immediate 的 substrate。但是它这两个就是你只要给 NR 或者给 NNM，然后你的 NAD 的也会增加，它就是这样的。就只要你这个 enzyme NM NMT 是没有问题的话，那么就会产生更多的 NAD，就是这样。
，OK OK。
嗯。所以就它现在就好像这个 market 也是比较多的吧。就是我们那个时候是问那个叫Cro 好像叫 Chromogen 嘛，就是问那个做 NR 的药厂是拿的那个药一开始，但是
OK。
就你也可以在外面买到，就是因为他们就卖很多这样。然后
那
好像会有很多，已经有一些研究是在人上面，它就是有没有对其他的，比如说代谢或者对运动的效率会有促进的作用，好像也有一些报道这样。
它这个调节线粒体的功能，应该来说是对运动这个有一定的这个效应的。
对对，应该是有促进的作用。就是他不知道现在有一些就是那些运动的产品，就是食品里面他们会不会加这个东西，有一些可能会有吧。
但是前一段时间我看新闻是国家药品食品监督管理局的是禁止了。
哦，
啊。前一段时间我看新闻是国家药监局的是把它禁止作为食品添加剂了。
哦，
它如果是药物的话，它如果认定成药物成分的话，它如果是已经
嗯
药物处方药、非处方药的里面那个成分，那它一般原则上应该不会再允许你在食品里面。
嗯，但是它其实
嗯，
也不是一个处方药呀，它是一个，就是也
它
就是
是国外的，把它
supplement。
作为食品
嗯，
添加剂噻。
对，
所以要
国外
就是
是已经有一些是
作为
做在食品里。
对啊，
supplement。
对对对，
嗯，但是我们国家它就是现在前一段时间是那个清单里面已经把它剔除了。
像这些 NR 这些分子的话，应该是可以穿过这个 BBB 的吧？可以进脑的，应该都问题不大。这都是小分子吧？这些东西。
这个算小分子，然后还有一个是它在那个如果是内皮细胞，它有个 extracellular 的receptor 的，就是它也会就是直接会去 metabolize 它，它也会有一个叫CD70几还是60几，对，它有一个 就
因为
是膜
它
上
它应该
面有一
是通过
个代谢的。
receptor 那个代谢
它有一它它
来
是这样
进行传递的。
这样转运的嘛？是的，是的。
对对，然后
嗯，
也能进细胞，就
嗯，这个蛮重要的。因为我们做牛肉酸的话，这个东西都要先想这个东西能不能
对
进，
对对对，那个对你
能
们比
不能透过
较重要，因
BBB？
为嗯。
嗯。
不要怎么考虑的。
好。
好好，
好，
如果大家没有什么参会的人没有什么问题的话，我们今天就非常感谢你们两位。
好，谢谢大
啊，
家，
谢谢张老师，谢谢。
嗯
谢谢
啊，
陈老师邀
好，
请，好，
好，谢谢
好，
谢谢你
好，
周末愉快。
周末愉快，拜拜。
嗯啊。